Hey guys, I promised that I would make more of an explanation and um, instructional video about the Gummy Bear Science Lab for osmosis. So here I am making it. um, okay, so this Gummy Bear Science Lab, the whole point of it is for you guys to witness or observe osmosis. So what you're going to be doing is in uh, 5 different cups, you're going to be maybe making different solutions. Um, one of them actually is just water, but different solutions to put a gummy bear in to see if leaving a gummy bear in the solution will change its shape. So here you have your purpose to observe the effects of osmosis on a gummy bear. And then you have your hypothesis, and it gives you 5 different ones for each of the cups. So it has the gummy bear left in plain water will shrink, swell, or stay the same. And I just want you to circle them. It's a Kami assignment, so you're probably going to be able to either use the drawing uh, thing or the shapes as well. Um, and then we have the materials, and you have 5 Dixie cups. You can use any kind of plastic cups that you have at home. Plastic preferable because you're going to be leaving this overnight with water in it, and with paper cups it might um, kind of seep through or something to that effect. And then you're going to have water. Now, you don't have to do exactly 2 tablespoons of salt or baking soda, and we're not going to be real scientific in measuring how much water we have. So, and then we need vinegar, 5 gummy bears, and then a ruler. And if you have a scale, great. If you don't have a scale at home, that's just fine. We will have a scale here at school, but if you're doing it this at home, if you don't have a scale, it's just fine. Um, and then you need a marker for labeling the cups, and you don't really need a measuring cup either. So what you're going to do is you're going to first label your cups. With one's going to have water in it, one's going to have salt water, baking soda water, just vinegar, and you're going to have an empty cup that you need to put control group on. Because if you remember in the beginning of the year, we kind of talked about how each experiment needs to have a standard that you go against. So if there's nothing to compare your experiment to, then your experiment kind of becomes invalid. So that's what our control group is for. So then you're going to put water um, in these cups. So obviously in the water cup, you're going to put water. Then in the salt water cup and the baking soda cup, you're going to put some water in there. Then in the saltwater cup, you're going to add 1 heaping spoon of salt. It doesn't have to be 2 tablespoons or whatever. 1 heaping spoon of salt in the water, into the water in the cup labeled salt water. And then you're going to stir it to make sure it's all dissolved and stuff. Then in the cup labeled baking soda, you're going to put a heaping tablespoon or just any kind of spoon of baking soda into the water, and then stir until it is dissolved. Then now you should have all of the solutions prepared. Oh, and then you're going to have another cup that just has vinegar in it. So add vinegar to the cup labeled vinegar, and that's just distilled white vinegar. Um, and then after you have all your solutions ready, you're going to measure and describe the 5 gummy bears using the scale and the rulers. Again, if you don't have a scale, it's fine. And you're going to record the data on the before in your tables. Now, if you scroll down here, you have a before and after. And before you put any of the gummy bears in the solutions, you need to write the color, the length, the width, thickness, and the mass. That's what the scale is for. So if you don't have a scale, you're fine. But you're going to measure these. And here it has the length, width, and then if you turn it on its side, the thickness. And then you're going to choose which gummy bear goes in what, and that's what these spaces are for. So observations and measurements of a gummy bear in salt water. So you're going to put something like that right there. And then you're going to place each gummy bear in the cup. And I recommend putting this on like a paper plate or a plastic styrofoam plate or something, and putting it somewhere where it won't be bumped or anything to that effect, because you're going to do this overnight. So then the next day, you're going to go back to your gummy bear cups, and you're going to carefully remove the gummy bears from the cups. I would suggest draining them first, but make sure that your gummy bears don't go down the sink. And then you're going to measure the after. So the length, the width, the thickness. Now, if, for example, if one of them is dissolved or just kind of mush, just put in the after dissolved. And that is data that you want to record. Then at the very end, you have this analysis and conclusion. And in your own words, you're going to describe the difference between the 4 gummy bears after the experiment. And I want you to remember to be specific and use details from your recorded data. So I don't want to just have it was in the water and then it got bigger. No, I want to hear something like I put the green gummy bear in the water cup and it increased in length 4 centimeters or whatever it may be. So stuff like that. And then I want you to tell me why you think this happened. So, for example, that green gummy bear in the water increased 4 centimeters because there was more solutes in the gummy bear than in the water. Something to that effect. So, I hope this helps, and I hope you guys are having a beautiful day.